All right. I think I will get started. I'd like to introduce our speaker for today. If you are, I think everyone is muted, but if not, we just ask that you try to stay muted during this. Certainly during the question and answer session, we encourage you to speak up. If I've muted somebody that can't speak, I fear like we have somebody that's called in. If you have an ability to type something to the chat box, I'll monitor that for questions and comments as well. So welcome. Today is James Kelly, part of his master's degree at the Auburn University School of Fisheries, Aquaculture, and Aquatic Sciences is his thesis work. And so he's going to present today on the effect of intertidal culture practices on the levels of Vibrio species in farmed oysters, Chrysostria gigas, from the Pacific Northwest. James came to the lab from Susquehanna University, where he earned his BS in biology in 2018. He also played defensive end there. So I have a teenage son who's been grateful for some of the football advice that he's gotten from James. And James began his Master's of Science with us in May 2019. He actually started a little bit earlier and came on board. And James is a product of the Memorandum of Understanding, the partnership between the FDA's Seafood Lab and the Dauphin Island Sea Lab. There's a partnership there of collaborative research, and James has been a part of that. Going forward, James has been applying, I know, because I've been writing letters of support and answering questions for folks, that he's interviewing for research positions, and we're working on getting his thesis worked up for a manuscript for peer-reviewed publication. With that, James, I will hand it over to you, and you should be able to share your screen.
All right, I'm gonna try and do that now. All right. All right, can everyone see title slide?
Yep.
All right, thanks, Bill, for that introduction. Like he said, I'll be presenting the work from my thesis on effects of those intertidal culture practices on Vibrio levels in farmed oysters from the Pacific Northwest. So oysters are traditionally wild harvested, and as we can see in these cases, they tend to form irregular shapes and grow in clusters, which are not desirable traits for the half-shell market. So even though this practice is still used with wild populations in decline, the practice of aquaculture has been growing as used to farm oysters. Oysters this way are raised single set, which improves their shape and may fetch a better price on the half-shell market. Oysters are the leading molluscan aquaculture taxon by quantity produced, and the industry had a harvest value in the United States of $186 million as of 2017. So to make it in this industry, farmers must decide which culture methods and practices are best fit for them. The oyster farms can utilize off-bottom methods, where oysters are grown elevated in the water column, typically suspended or floating. And they can use on-bottom methods, where oysters are grown directly on the substrate, closely mimicking a natural reef. Farmers can develop a variety of pre-harvest handling techniques as well, including tumbling, refrigeration, and desiccation, to help increase the overall quality of their product. Desiccation or air drying is important in controlling biofouling to prevent unwanted organisms from establishing, like barnacles. And the process is controlled by the farmer in places such as the Alabama coast, where tidal shifts are usually shorter. Here, farmers will manually desiccate their oysters for about 18 to 24 hours. But this process can also occur naturally, as within the Northeast and the Pacific Northwest, where large tidal shifts expose the entire tidal flat each day. This allows for all oysters to desiccate, including those on bottom, and can be referred to as tidal desiccation. This happens on a daily occurrence where these intertidal farming practices take place. So being that oysters are consumed raw, seafood safety is important to consider. Oysters are bivalve mollusks, which means they feed by filtering, the water surrounding them, which may include any microbes in the water as well. And of these microbial bacteria, one of them is from the gene Vibrio. There are two specific species we looked at in this study that are considered a public health risk. The first one is Vibrio parahemolyticus, or VP, and it is the leading cause of seafood consumption-related infection, and that infection can cause gastroenteritis and septicemia. The pathogenicity of VP has been correlated to the presence of either the TDH and or the TRH gene, but these are not the only markers of pathogenicity, but common ones used in methods of detection. So Vibrio vulnificus is the second species. Those who are immune compromised are at the highest risk for infection from consuming raw seafood. And this can lead to gastroenteritis and primary septicemia. It is an important pathogen in seafood safety because it can account for 95% of all seafood-borne related deaths. These Vibrio are naturally occurring in waters where oysters are grown. Therefore, we cannot see them as a risk of contamination in oysters. Vibrios are commonly associated with outbreaks from eating raw oysters, especially the half-shell market, with some of the more recent outbreaks coming from shellfish where intertidal farming practices are common. So farmers must be aware of these Vibrio risks during the farming process. Vibrios are naturally found in waters, so there is what we can consider ambient or background levels within them when oysters are actively filter feeding. When farmers perform routine handling practices, such as desiccation, oysters are removed from the water and will clam up in order to conserve moisture. And the elevated temperatures, along with direct sunlight and the lack of filter feeding, may cause the rapid growth of Vibrios within them. So after handling practices are over, the oysters are returned to the water or resubmersed, in which filter feeding can resume. And this allows oysters to flush out these increased levels of bacteria over time. And depending on the area and region, there are specific times allowed which are deemed necessary to observe levels decrease to what is thought to be ambient or background. So the effects of common culture practices on Vibrio species are important to study. In Alabama, a study conducted by a previous student at the Auburn Shellfish Lab on the effects of desiccation on Vibrio species levels, as well as ploidy and how they may have affected them. Ploidy in oysters being, triploid, diploid. We also looked at different pre-harvest handling practices in our group as well and studied how they affected Vibrio levels, as well as common culturing methods and gear types. And we also looked at geographic location and how they compare to studies we've done here in the Gulf of Mexico. So these studies we looked at have mostly looked at how manual desiccation has affected Vibrio levels within Crassostrea virginica, the eastern oyster, is the primary oyster aquaculture species of the area. But for our study, while we wanted to stay keyed in on the idea of desiccation affecting these Vibrio levels, we wanted to look further at geographic location in areas where intertidal farming practices with multiple culture methods are common, to see if oysters and associated Vibrio levels respond differently. So in Washington State, specifically in the greater Puget Sound, there are two main types of culture methods used. On bottom, or beach culture, has been the traditional means of oyster production in the region, and oysters here are left to grow directly on the substrate. Flip bag farming, or also referred to as tumble bag, is relatively new to the region. With this technique, a rope runs 2 feet off the ground with mesh bags hanging from it to house the oysters. Attached to the bottom of the mesh bag is a flotation device, which will cause them to rise and fall with the tides. And the movement caused from this will allow new shell growth to break off, and the constant tumbling creates a firmer meat and deeper cupped oyster, which are all desirable traits in the half-shell market. Both of these methods are subjected to the large tidal cycles, leaving farming areas completely void of water for up to 7 hours a day. Here they are primarily farming Pacific oysters or Crassostrea gigas, and comparatively to its sister species Virginica, it does have distinct ribs on its shell and a lighter pigmented abductor muscle, but overall they're very similar to each other. And currently in Washington state, farmers must wait until 4 hours of resubmersion from the incoming tide is reached until harvesting their oysters. With that in mind, we had a twofold objective for the study. The first one was to analyze the effects of tidal desiccation and resubmersion on the levels of Vibrio species. And our first hypothesis was that after exposure to elevated air temperatures during low tide, levels of Vibrio will be significantly elevated from levels before air exposure. So to test this, we took pre-exposure or T-pre samples right as the tide went out, exposing the oysters. This would give us initial Vibrio levels. And then we took post-exposure or T-post samples collected right before the oysters were resubmerged by the tide, about 7 hours later. Our second hypothesis was that after tidal desiccation, 4 hours of resubmersion will result in Vibrio levels decreasing and returning to those background levels. So to test this, samples were collected 2 hours after resubmersion, or T2, and 4 hours at T4 to monitor the rate at which Vibrio levels were fluctuating inside these oysters. The last samples would be collected 24 hours after the first samples were collected at TF. So our second objective was to compare the effects of the two culture methods on Vibrio levels. And it was hypothesized that the recovery times required for elevated Vibrio levels to decrease and return to background levels will not be significantly different between the two culture methods. So within the Pacific Northwest region, Washington State was chosen as the site location. Specifically, within the Puget Sound, a commercial shellfish farm was used just off of Samish Island. The experiment totaled 5 trials and was conducted during the summer months of 2019 and 2020 to ensure Vibrio risk was elevated to its highest point. Commercial flip bag long lines were utilized, and flip bags were used with those specific white buoys that we see, which made them easier to locate and distinguish from the farmer's gear when tides were high. For beach culture, oysters were selected at random from the on-bottom farming site, and those to be collected during high tide were placed in harvest bags with those white buoys attached to them. Triplicate samples were collected at each time point for each culture method. So this meant that 6 samples altogether were collected at each time point, totaling 30 samples collected for the whole trial. So samples were tested for 4 separate Vibrio targets, Vibrio parahemolyticus, or total VP, the pathogenic strains that we had mentioned previously, TRH plus and TDH plus, and Vibrio vulnificus. Vibrio bacteria were enriched overnight with a 3-tube most probable number analysis. And after this overnight enrichment period, those tubes that showed bacterial or turbid growth were sampled, heated to 95 degrees Celsius to extract the DNA. They were then frozen and shipped back to our lab here on Dauphin Island. So back on the island, real-time PCR analysis was then performed to identify and confirm the presence of each Vibrio target. The number of tubes that were positive for each target were then used to calculate the most probable number of bacteria per gram of oyster using this table. So prior to statistical analysis, Vibrio data was logged. The data was analyzed per trial using generalized linear models in R testing. And with 4 Vibrio targets times 5 total trials, there were 20 total Vibrio target trial combinations. And with the 2 culture methods for each combination, made for 40 total comparisons. So this set of graphs will show the levels of total VP from each trial. On the vertical y-axis, we see that levels of Vibrio are represented as logs MPN per gram. And on the horizontal x-axis, we see the sampling times tested in chronological order, starting with oysters tested prior to tidal desiccation, following maximum exposure, 2 hours of resubmersion, 4 hours, and 24 hours from original pre-exposure collection, representing a full tidal cycle. So first, looking at how levels of total VP changed following maximum exposure. In trial A, levels increased in both methods by over a log in this trial. And then we saw similar increases for both methods observed in trial B, C, and E. Although in trial D, we did see decreases in beach culture oysters for this target, with only a small increase in footbag oysters. So in comparing how total VP levels changed then following resubmersion, remembering that 4 hours is the industry standard for the region, we saw decreases generally viewed for both methods in trials A, B, and D, with levels also returning to near T-pre. Although levels of VP were observed to increase in beach culture oysters in trial C and flipbag oysters here in trial E following this resubmersion. And even though in both trials the other culture method saw decreases, these levels were still higher than those observed prior to resubmersion at T3. So in comparing both culture methods to each other, in trial A, we see that flipbag oyster levels are higher at every time point sampled, although fluctuations from each time point are similar between them. This is also the case in trial B, although there is that noticeable difference we see at the TF time point where flipbag levels are higher. In trial C, we also see that flipbag levels are generally higher, aside from levels at that T4 time point. And this was also interesting as levels in beach culture oysters increased following this resubmersion when they slightly decreased in flipbag oysters. But similar to trial B, we did see this increase at TF when beach culture levels decreased. So in trials D and E, while some noticeable differences occurred for levels in both beach culture and flipbag oysters, variation in the two trials between time points is prevalent, making generalizations difficult to make. But overall, total VP levels in flipbags did appear to be higher than in beach culture oysters when trends were noticed. So moving on to the pathogenic TRH plus strain. We see here that in trials A, C, and D, a general increase was observed in both culture methods following maximum exposure. In trial B's beach culture oysters, and then in trial E's flipbag oysters, we did see decreases following maximum exposure, with increases in the opposite culture method for both trials. So then comparing how these levels changed following resubmersion. For trial A, from both methods, we saw levels decrease following that 4 hours of resubmersion to near T-pre levels. And we saw levels in beach culture oysters in the remaining trials unexpectedly stay about the same. In flipbag oysters following 4 hours of resubmersion, while levels did decrease in some trials, it was only minimal. Although in trial B, we did see levels decrease to those T-pre levels observed. In trial D, those oysters in flipbags stayed, levels in oysters and flipbags stayed about the same. But in trial E, we saw a dramatic increase in TRH plus levels following resubmersion. And in both of these trials, in trials D and E, we didn't see levels return to T-pre. So in comparing both culture methods, in general, levels of TRH plus VP in oysters were higher in all trials compared to levels in flipbag oysters. We did notice one difference in trial B, in trial E, but that was it for among trials. Any significant difference? In trial C, resubmersion did seem to affect those beach culture oysters differently as levels increased after 4 hours, just like in total VP, as opposed to the flip bag oysters where levels decreased. This next set of graphs will show the levels of pathogenic TDH plus from each trial. Before starting, it is important to note that levels were generally lower, with about a third of all samples not detecting any TDH plus. Although we did see levels of pathogenic TDH plus increase following maximum exposure in all trials, but was most noticeable in trial C, where TDH plus was detected in most of the samples. So following 4 hours of resubmersion, levels did decrease in that trial C for both culture methods. And in general, levels decreased or stayed the same following 4 hours of resubmersion, like as we see in trials A, B, and D. And they stayed relatively low in relation to other Vibrio species, but in cases where levels did increase, such as in trial E here, levels at T4 were still pretty low. So the low limits of detection made for difficulty in locating trends altogether. So in comparing both culture methods, in trial C, levels of pathogenic VP-TDH plus were observed to be higher in beach culture oysters than those in flip bag oysters in all time points, other than T-pre. In trial E, while levels stayed relatively low throughout the trial, the differences at T2 and TF between the two is worth noticing, as levels in flip bags were higher than those in beach culture oysters. In the remaining trials, all levels were relatively low, combined with odd variation among sampling times, making trends in comparisons difficult to see. But for this Vibrio target, we did notice that beach culture, the levels in those beach culture oysters were higher. So for Vibrio volnificus, or VV, it's again important to note that the majority of samples throughout the study did not detect any VV, as you can see from most of the trials, having little to no change in levels in many time points. Because of this, statistical analyses were not run for this species. But for trial A, we did notice changes among time points. And in this trial, levels did increase following maximum exposure, dramatically so in those beach culture oysters. And while there were some increases, they were minimal, and changes only did seem to occur in levels from those oysters in flip bags. So following resubmersion, trial A did see decreases through that 4 hours of resubmersion to levels seen near T-pre. Decreases were also observed in flip bag oysters for trials B and E. But in trials C and D, we saw increases again after 4 hours of resubmersion, with beach culture oysters for the remaining trials showing relatively low or staying constant throughout. But overall, the trials showed variation similar to TDH plus, and because samples because there were samples with no detection, it made for difficulty in locating trends. So in comparing both culture methods, in cases where time points did view to be different, levels were increased in flip bag oysters in just about every case, especially in the trial C and D at the critical T4 time points. So to refresh, our first objective was to analyze the effects of tidal desiccation, and we did hypothesize that Vibrio levels would increase following maximum exposure. So after exposure to elevated air temperatures, we did notice that Vibrio levels increased in 82% of those trial Vibrio target culture method combinations. And while these increases were not always statistically significant, they do indicate a trend in the data of elevated levels. Our first objective was to also analyze the effects of resubmersion following tidal desiccation. And we did hypothesize that Vibrio levels would decrease from elevated levels and return to those observed prior. So following resubmersion, in those combinations where levels increased following tidal desiccation, we saw decreases in 64% of cases. And again, these decreases were not always significant, but indicate that a majority of cases that saw increases then saw decreases following the industry standard of 4 hours of resubmersion. And of those 33 cases, we saw that 36% of the time, Vibrio levels were able to decrease to those levels seen at T-pre or below. So it is also important to note that Vibrio targets Vibrio vulnificus and the TDH plus strain were low throughout the study, making generalizations difficult to come by. But the results are consistent with existing work as Vibrio parahemolyticus, specifically the TRH plus strain, are found in the majority of illnesses. And in the state of Washington, there has not been a reported case of vulnificus infection from consumption of raw shellfish. Yeah. So the second objective for the study was to compare the effects of the two culture methods on Vibrio species levels. And when comparing the two culture methods, in general, levels of Vibrio species were higher in flipbag oysters than in beach culture oysters. And in cases where the effective culture method was significant, levels in flipbag oysters were higher than in beach culture oysters in all trials but one among targets. So this could be due to disturbances in the water column affecting the oyster's behavior within the flip bags, such as wave action caused by recreational activities or marine mammals. But this is all just speculative and we need to be further investigated. So of the 20 trial Vibrio target combinations for each culture method, increases in Vibrio levels were observed in 75% of cases for beach culture oysters and 85% of cases for flipbag oysters, meaning levels were able to increase more frequently in oysters from flip bags. And then following 4 hours of resubmersion, decreases in levels were observed following those levels that increased at T-post in 93% of beach culture oysters in cases of beach culture oysters and 71% of flip bag oyster cases. We also saw that in 60% of the beach culture oyster cases that these levels returned to those observed at T-pre, and this was the case in only 47% of flip bag oysters. So even though it's only a slight difference, there is a small trend noticed where beach culture oysters were able to recover from elevated Vibrio levels in more cases than flip bag oysters. So in summary, in these intertidal culture practices following maximum air exposure, during tidal desiccation, Vibrio levels did elevate in oysters. And then following resubmersion from the incoming tide, 4 hours of resubmersion often allowed for those elevated levels to decrease, and in cases similar to those levels observed prior to tidal desiccation. In comparing the two culture methods, in general, Vibrio levels in oysters grown out in flip bags were higher than those levels in oysters grown on bottom. And elevated levels in oysters from flip bags did not recover during resubmersion as frequently as oysters from beach culturing. With that, I'd like to give a special thanks to the Dauphin Island Sea Lab and FDA Fellowship Program for making all this research possible. The entire staff at the FDA and the Auburn Shellfish Lab also deserve a huge shout out for their continued efforts during the past year in such trying times that were brought to us. Out in Washington, Taylor Shellfish allowed us to use their equipment and farm to make this study possible, as well as Dr. Roberts, who let us invade his lab space for the time out there. A special thanks also to Vivian Tucker, Rob, Kendall, and Sam, who all dedicated their time and efforts to getting those samples processed safely. And with that, I will take any questions.
All right, do we have any questions in the room?
Hey James, this is Blair. Great talk. So I think the question that I have is why do you think that there is such a difference between the beach culture and the flip bag? Do you think it has something to do with like the flow rate through the flip bags, or is it potentially, you know, something with the contact with the sediment is allowing the Vibrio to kind of go back into a sediment environment? Do you have any ideas about why those differences exist?
Yeah, great question. So looking at how the oysters differ in the two methods, you know, they're exposed to sunlight in different ways. You know, on the beach, they're laid out directly on the substrate. So, you know, it's direct sunlight on them. And when they're in the flip bags, they are housed and they are all, you know, crowded in there. So maybe stocking density might have something to do with that as well. But I think that the biggest factor worth investigating further would be how wave action may affect how the behavior of these oysters might be affected. Maybe they're closing up more often and not able to, you know, flush out bacteria as often or as frequently as those on the beach. Yeah, we do have a student here working on effects of wave action, so maybe we will get some data on that. But I think that would be the biggest thing to investigate as to why we might be seeing this. Okay.
I'll keep the question train rolling here.
Sure.
Can you hear me okay, I'm assuming?
Yeah.
Okay, can you go to your slides where you had the graphs going over time?
Yeah, hold on.
You just closed it down, didn't you?
Yeah.
I'm sorry.
Okay. Okay. You asked me, you wanted to see where again? I'm sorry.
Doesn't matter, any of those ones where it's going over time.
Head back.
Great presentation, by the way.
Thank you. Appreciate that.
You're welcome. Yeah, one of these guys. So can you explain this to me a little bit in detail on the statistical approach that you take for this, just for curiosity?
So in terms of a statistical approach, so we're looking for significant differences, you know, from these levels seen at, you know, T-pre compared to, you know, following resubmersion at T-post. So altogether, we tested to look for an interaction between culture method and sampling time. And in about half of cases, we saw that there was a significant effect of culture method. And in those cases, flip bag oysters were higher altogether. But you know, looking forward into a publishable study in cases where we did see significant interaction, we will, from a statistical standpoint, look to do post hoc wise comparison tests where we can show with subscripts above the bars where, you know, levels were similar to each other. So for example, we would see like beach coasters at T-pre and T2 are most likely similar to each other, unlike levels at, you know, beach cultures and T-pre, and flip bag oysters at T2 would be significantly different. So what we're looking for is to see that there is a significant difference in those levels from T-pre increasing to T-post so that they're higher. And then we're seeing that levels then decrease through resubmersion, looking for similarities between levels at T4 compared to T3.
Okay, that is a very nice overview. I think that was good because I looked at your graph and you had on the right, you had letters represent significant differences, and I didn't see those. So you probably removed them for clarity.
Yes, yeah, because only only about 2 out of specifically trial B and E for this one, we did see that, but we did remove those for trial.
And it does appear like all these traits did have significant interactions opposed to them, eh?
Yeah, so on only these, I remember for this species and only trials B and E, there was that interaction. But the
Okay.
rest, we didn't see it.
Super. Okay, thank you very much for that clarity. Thank you.
Yeah, thanks for pointing that out.